Right, welcome everyone, wherever you are joining us from, to today's Global IMMUNOTALK. Now, this is a great initiative that was started by Carla Rothlin and Elena Zuniga, and that brings all aspects of immunology to a global audience from all corners of the world. And we rely on the goodwill of stellar speakers from all around the world who agree to give these talks. And today we welcome David Tarlinton. He joins us all the way from Australia, and in fact from the middle of the night, because it's 2 a.m. In Melbourne. David is originally from Sydney, Australia. He studied at the University of Sydney before he moved to Stanford to do a PhD with the Herzenbergs. And he then did postdocs with both Klaus Rajeski in Cologne, Germany, and with Gus Nossel at the Walter and Eliza Hall Institute in Melbourne. And actually, David stayed at the WEHI. In 1997, was appointed a laboratory head at the Walter and Eliza Hall Institute. And he actually stayed there until 2016, when he moved to Monash University, still in Melbourne, where he is professor and head of the Department of Immunology. Now, David's work focuses on the development and differentiation of B cells, and he has made key contributions to our understanding both of the germinal center reactions and the development of plasma cells and of memory cells, memory B cells. Now, as usual, today's talk will be followed by a Q&A session, which will be done offline in Twitter/X, in which David will endeavor to answer your questions over the next couple of weeks. And instructions on how to post your questions will be displayed at the end of this webinar. So in just a minute, I'm going to ask David to deliver his lecture, but as is usual in this series, I will start with a question before his talk. And the question is, what advice, David, would you give to your 25 to 35-year-old self?
Thanks, Kashana. Yeah, I think I would tell myself, if I could, to just relax a little bit and enjoy the moment. I think I tended to be incredibly stressed at that time in my life about what was going to happen in the future, and really failed to appreciate all the things that were going on around me, not necessarily that I was doing, but that were happening in my immediate environment. And I think I could have probably learned a lot and possibly enjoyed that interval much more if I had just managed to somehow take a breath every now and again. Not that I was, completely fixated on work, but I always seem to be doing PCRs, southern blots, whatever. Yeah, and not really taking advantage of, there's incredible circumstances in which I think many of us find ourselves during our career, and only really realize it in retrospect. That was perhaps the biggest disappointment, not seeing what I was actually participating in.
Well, these are wise words with the benefit of hindsight. So thank you, David. And I will now invite you to start sharing your screen and deliver your lecture, which is entitled Identifying the Mechanisms and Attributes of Plasma Cell Longevity.
Is that in the right format?
I think you need to swap the displays. I think in your presenter mode.
It's either that or I'm on mute. It's one of the two. There we go. How about that?
That's great. Thank you.
Okay. I might just get a laser pointer as well for this. Okay. So shall I start?
Yes, please.
Okay, fantastic. Well, thanks everyone for joining me at what hopefully is a much more salubrious time of the day than it might be for me. I'd like to talk about work we've done over the last few years on, as it says in the title, determination of plasma cell longevity, a question that has been, and a topic that has been at the forefront of my research over the last several years, and perhaps as a natural progression of work we started many, many years ago on germinal centers. So to put this into some kind of context, perhaps, overly emphasizing the importance of it, we can start perhaps with the idea of vaccination and why the persistence of antibody is important. So everyone, I think, is familiar with this process developed by Edward Jenner in, as it says here, 1796, at which he determined how it was possible to protect humans from smallpox infection by exposing them to to a variant of that particular virus. In this particular case, there was no attenuation of the virus. He basically just took a bet on this and gave James Phipps cowpox, and then did a test with N=1 by exposing him to variolous material, and no disease developed, which was extremely fortunate. And then, in many respects, the science of vaccination, or the phenomenology of vaccination, was born. And it took 100 years or more for immunology to catch up to this translational outcome, but it certainly emphasizes the value, if you like, of not only vaccination, but of the persistence of antibody, as it turned out. So if we now put this into a more contemporary context, and this is from a review of Andreas Radbruch and Thomas Dorner, where they showed the stages of the immune response to infection with a cytopathic virus, which is more or less what was happening in the case of James Phipps. And you can see the various lines being traced out on here, the components, for example, the virus. I'm sorry about that. I'm getting control of that laser pointer. Here we go. The cytopathic virus increases and diminishes as the immune response takes hold. Various forms of that response increase in frequency and then diminish, CD8 T cells and CD4 T cells. And you can see in this particular example, the persistence of neutralizing antibody. And this dashed line is meant to indicate the amount of antibody necessary for neutralization. So in this prototypical example, one can see how single exposure to a virus or a vaccine can create lifelong protection mediated by the persistence of neutralizing antibody. And of course, there are clearly other components of the immune system that are persisting as well. So how is this achieved? What is the nature of the cells that are able to continue to persist and to produce this antibody over such extended periods? In this case, in this example, over 75 years. Is there something unique about them, or is that a general property of all plasma cells and all immune responses? What are the criteria by which such cells are created, segregated? Where do they live, and how are they sustained? Those are the kind of questions that naturally occur to one when looking at this kind of data. So, as I said, my interest in plasma cells arose from working on germinal centers, and what's shown here is a schematic that I've used for the last 20 years, I think, to describe the process of an immune response. In this instance, antigen introduced is able to activate a B cell and, of course, separately a T cell. The B and T cell combined, specific for the same antigen, will initiate a couple of different outcomes. One is that there will be the formation of antibody secreting cells immediately, if you like, upon this interaction of B and T cells and proliferation. There's a plasmablast, so still proliferating cells that secrete antibody. And these cells, which sit outside the B cell follicle in the secondary lymphoid organs, typically have a short lifespan, such that they are dead within 1 to 2 weeks of their formation. So this provides an early but transient production of antibody specific for the challenge, which initiated the entire process. Also, out of this initial interaction and early B cell proliferation, some of those B cells will stop proliferating and yet remain alive and in circulation, and they'll be a form of memory cells reflecting the initial affinity of the B cells that were activated in the first instance. But importantly, for the efficiency and efficacy of our immune responses, some of these B cells and the T cells will migrate back into the follicle and form germinal centers. Now, I'm sure there'll be several talks in the series on the properties of germinal centers and the activities within them. But for the purposes of this talk, I'll just highlight a couple of things. One is that there's somatic mutation and selection of the B cells within the germinal center to improve their affinity for the immunizing antigen. And this occurs over multiple iterations of proliferation, mutation, selection. with corresponding amounts of cell death for those cells, B cells that fail to improve. But those that do improve are then sent through the process again and again, and in this iterative process, improve their affinity. Out of this process arise two populations, again, memory cells with improved affinity relative to the initial pre-germinal center memory cells and the initial activating B cell. And these will be available for recall if such a process is necessary, which we might get to in some subsequent part of the talk. But the basis of this talk, there will also be B cells emigrating from the germinal center that are committed to or already are secreting antibody in the form of plasmablasts. And these cells will reflect the affinity improvements that have happened within the germinal center. So the antibody they secrete will be of a higher affinity than was achieved or was present in the antibody produced by those initial plasmablasts that arose at the very first few days. And from these plasmablasts, they'll stop proliferating, and that will make them, by definition, plasma cells, so non-proliferating antibody-secreting cells. And these have the capacity, will be immature at the outset, but they have the capacity to mature and to persist in that state. And that mature plasma cell has the opportunity to persist in locations specifically in the bone marrow. There are also long-lived antibody-secreting cells or persistent antibody-secreting cells in other locations such as gut, but the purposes of this talk, we're going to focus mostly on the bone marrow. So this schema describes antigen introduction, B cell activation, and a T cell dependent immune response, giving rise to short-lived antibody-secreting cells that provide low affinity antibody in a transient fashion. But this transient antibody is supplemented and then ultimately superseded by antibody that comes from antibody-secreting cells that have originated from throughout the affinity maturation process of the germinal center, that are then able to be to mature and to persist in that state of antibody production of high affinity antibody over an extended period. So that's the overview. We're now going to look at some of the details within this. If one wishes to study and analyze and determine what are the attributes of these cells that are persisting and secreting antibody over this extended period, of course, one has to identify them. Seems like it should be simple, but of course it was not. It relied, well, from my perspective anyway, on what was a terrific innovation at the time, some 20 years ago, when Stephen Nutt at the Walter and Eliza Hall Institute introduced a GFP reporter into the locus encoding BLIMP1 protein. So the locus is PRDM1, the protein is BLIMP, and it at that time was considered a master regulator of plasma cell differentiation. It's certainly critical for that, and it's expressed in all antibody secreting cells, but one could debate whether it really is the master regulator. It certainly comes on very early and is expressed in all antibody secreting cells. So as a result of that property, all antibody secreting cells in these gene targeted mice express GFP of one of the alleles of the PRDM1 locus at the point at which they differentiate and start shortly after the process of differentiation and the initiation of antibody secretion. And what you can see in these histograms below is what that looked like in the animals in which this manipulation was done. One can see populations of GFP positive and Syndican positive, CD138, there's a new nomenclature for that, cells sitting within the spleen, the blood, and the bone marrow. And in the histograms beside that is the intensity of BLIMP expression in those antibody-secreting cells. And the fact that they were antibody-secreting cells is shown by ELISpot analysis of those isolated cells on the right-hand side. This is published a long time ago, so I'm not necessarily going to go into much detail about it. And those cells, could be seen as GFP positive, but what was apparent immediately, was the amount of GFP expressed in those antibody-secreting cells was varied. So within the spleen, for example, there were two apparent populations determined by BLIMP expression, high and intermediate. In the blood, all of the cells were intermediate. And in the bone marrow, the vast majority of the cells were high. So there was variation in BLIMP expression, already revealing heterogeneity amongst antibody-secreting cells, even just with this one parameter. And a heterogeneity that was also, to a degree, predicated on location. So that suggests that there may be differences in the properties of these antibody-secreting cells based on that separation by BLIMP, and that indeed turned out to be the case. So this is a very old-fashioned experiment looking at lifespan, and it was to use incorporation of the nucleotide analog BRDU into the mice over periods of 7, 14, and 28 days, and then assessing the incorporation into the plasma cells that were separated according to the amount of BLIMP. And not to go again into any details, but just the fact that many of the GFP or BLIMP intermediate plasma cells incorporated BRDU within 7 to 14 days indicated that they were a rapidly proliferating and turning over population, whereas the cells that expressed, plasma cells that expressed high amounts of BLIMP showed much less incorporation of BRDU over the same period, and in fact it plateaued, which suggested that that population comprised, to a degree, long-lived cells, so cells that didn't have to be replenished within the 4-week period that we were doing the study. And below is some supporting information of DNA content. So even at this early stage, it suggested that by looking at those cells that express the higher amounts of BLIMP, plasma cells that express the higher amounts of BLIMP, we may have, if you like, isolated and identified those cells that were long-lived. And of course, we went on to do RNA sequencing based on the segregation. Now, thinking that we had identified mature and immature or long-lived and short-lived antibody-secreting cells, and looked at the gene expression program. And again, this was published about 10 years ago, to see if there were indicators that correlated with the maturation and the other properties of those antibody-secreting cells, such as persistence and location. What's shown in this heat map, comparing all different types of B cells along in each column, which you can see from follicular B cells, marginal zone, B1, germinal center B cells, and then 3 types of plasma cells, 2 from the spleen and 1 from the bone marrow. And looking at genes that differ across that spectrum, you can see that all of the B cell types, which are the left-hand side of each of the panels, look reasonably similar, and all of the plasma cells look reasonably similar to each other and quite different from the B cells. And on the left-hand panel, it's the genes that were most downregulated in the transition from a B cell to a plasma cell. And on the right-hand side is the 50 genes that were most upregulated in that same transition from a B cell to a plasma cell. The point I'm trying to make is two things. So one is that there's a huge change in the transcriptional program of B cells as they become plasma cells. And the second thing is that the plasma cells that we had identified as being different based on BLIMP were actually reasonably or very similar in many of the other genes and all the other genes that they expressed. So there wasn't as if there was a huge change in the different stages of what we perceive to be development of plasma cells associated with the RNA that we were measuring by RNA sequencing. In fact, they all look pretty similar, and yet we could clearly identify different properties for them, not only the increase in BLIMP expression, but also their location and their turnover. So there was no obvious indicator from the RNA as to what the properties were that permitted a cell to be in the bone marrow and to persist and to have a long life compared to those that were in the spleen and didn't have such a long life. So if I could summarize this to an extent, we'd identified at this time that there were immature plasma cells and mature plasma cells. And in our case, it was determined by the amount of BLIMP that they expressed. There were other schemes for segregating plasma cell behavior available at this time, and a lot of work done by many other groups, some of which are referenced in the lower left-hand side of this slide, contributes to this kind of overview that I'm talking about. So immature plasma cells were considered to turn over rapidly, whereas mature plasma cells did not. Mature plasma cells were considered to secrete large amounts of antibody per cell per unit time compared to immature plasma cell. So function was improved. there were considered to be changes in markers, such that mature plasma cells had a particular distribution, and some of those are listed here of beta 20, class 2, CD19, and so on. Mature plasma cells were thought to live in the bone marrow and the lamina propria, whereas the immature plasma cells were considered to be in the peripheral organs, predominantly spleen and lymph nodes, in mice at least. Isotype pattern was considered to be different, so immature cells were considered largely to be IgM dominating over IgG, whereas mature cells, it'd be the class switch isotypes dominating over IgM. These cells were thought to have different metabolism based on glucose uptake and differences in autophagy and mTOR activity. And also, long mature plasma cells were considered to come from the germinal center, whereas immature were typically considered to be the product and the end state of those extrafollicular responses. So this might say there's two types. There's an immature cell and there's a mature cell, and a plasma cell simply has to transition from one to the other in order to achieve longevity, persistence, and continued antibody production. And that would be the explanation for the persisting antibody in the case of the infection and the vaccines that we were discussing at the very beginning. And that would be able to be depicted in a scheme like this if one is to consider lifespan across the bottom and the percent of cells persisting. So there would be immature cells, the majority of which were short-lived, so would decay very rapidly down to a point of 0, and then a long-lived compartment, which may be quite uniform and would show a lifespan decay considerably different to that of the short-lived cells, but relatively consistent within those cells that will mature. So that would be a dichotomy, if you like, between those two states. But there's an alternative, and the alternative is that, in fact, it's a continuum of lifespans. So there are short-lived cells and long-lived cells, and any individual cell has the potential to be placed at any point along this spectrum based on attributes that are yet to be determined. So in the one case on the left, you could imagine there's an actual differentiation state that goes from point A to point B. And the other, it's more of a spectrum, and there may be many more gradual changes and many more different circumstances that can dictate lifespan as one place and another. One of them could be, for example, the random assignment of lifespan at the time of formation. The other could be where the cell actually winds up and the environment in which it sits and how nurturing that is, if that turns out to be the case, for the maintenance of that plasma cell. So what do we know about the survival of plasma cells, about their persistence? What mechanism is there? What molecules are involved? And how do we begin to disentangle all the different components that may contribute to where the cell sits in this lifespan spectrum? And do we even know which of these models of plasma cell lifespan is in fact correct? So to talk first about the molecules of survival, and this will be a little bit important later on. We were interested for many years in the role of the molecules that regulate cell survival, so the pro and anti-apoptotic molecules of the BCL2 family, and their role in B-cell immunity, particularly in selection in germinal center responses, and as I'm about to show you, in plasma cell persistence. And there are a number of molecules that are pro-survival, shown here on the left of this seesaw, and others that are pro-apoptotic on the right. And cell survival depends upon achieving a balance of and an excess, as it turns out, of the pro-survival proteins over the pro-apoptotic ones. And through a range of experiments that I'm not going to go into, in examining what was necessary for plasma cell survival, we had ruled out that BCL2, XL, and W had any role. And instead, all the indicators were that MCL1, a very short-lived pro-survival protein, was going to be the determinant of plasma cell persistence. We confirmed this in what at the time struck me as quite emphatic fashion by creating mice, and I apologize for the complexity of the schematic, but we did it by bone marrow reconstitution to create animals in which we could inducibly delete both alleles or one allele or no alleles of MCL1 from all of the B lineage cells in these animals by the introduction of tamoxifen. So it was a tamoxifen-inducible CRE, which acted on a floxed allele of MCL1, and we had mice in which both alleles of MCL1 were floxed, just at the top. One allele of MCL1 was floxed, or no alleles of MCL1 were floxed. And 2 days after sequential doses of tamoxifen, we examined the mice to determine the status of their antibody-secreting cells. And what we found was really quite striking and really clear that if we deleted both alleles of MCL1, which is shown at the bottom, then all plasma cells, which would be sitting in these 2 pink boxes here or here, were effectively deleted from these mice within that 24-hour period after the second gavage. So pretty unequivocally, from this data, it was concluded that MCL1 was the dominant survival factor needed to sustain antibody-secreting cells. But what I mentioned, and I'll show the summation of that over many different animals and circumstances, and what you can see in the white bars in these 2 right-hand columns are the reduction in antibody-secreting cells of both plasmablast and plasma cell forms after the deletion of 2 alleles of MCL1 in bone marrow and spleen. So no matter where we looked, the antibody-secreting cells were gone on the deletion of these alleles, clearly indicating that this was the key survival factor for all of the antibody-secreting cells, or the vast majority of them anyway, in these mice. So if one looked at this scheme of antibody-secreting cell production, where it's coming from the germinal center, what we just shown was that throughout this set of experiments, that arising from B-cell activation from, in this case, the germinal center, immature plasma cells had the capacity to give rise to short-lived and long-lived antibody-secreting cells that were distinguished from each other, at least in this circumstance, by the amount of Blint that they expressed. They existed in the spleen. Some migrated through the blood and took up residency in the bone marrow, where they were also able to persist for extended periods. But all of these cells, irrespective of where they were or what the maturation state was, were dependent on continued expression of MCL1. And I mentioned a little while ago that MCL1 itself is a very short-lived pro-survival protein. So in order to be able to sustain these cells over an extended period, it itself has to be continually expressed, which means that the signals that stimulate production have to be continually present or episodically present, at least at a frequency able to sustain that amount of MCL1. So that's the kind of clue as to what's necessary to sustain plasma cells. They need MCL1, of course, but then they need the signals that will drive MCL1 expression. And those signals derive from what's considered now as the plasma cell survival niche. So this idea was championed by Ian McLennan as a way of explaining the fixed number of plasma cells, the homeostasis of the population. But that idea was advanced by others, particularly Andreas Radbruch, to indicate that this was a system whereby not only would the cells be anchored in one point, but they would be given access to the array of signals that they would need. The plasma cells would be given access to the array of signals they would need to sustain, as we just demonstrated, the expression of MCL1 and therefore their survival. So the niche is still, to a degree, a concept rather than a concrete collection of cell types in every circumstance. It's known that there must be a nucleating cell which expresses CXCL12, the ligand for CXCR4 that all of the plasma cells express, that brings them to this location. It brings other hematopoietic cells into the same orbit, and many of these cells, all of the ones shown here, for example, are secreting factors which are non-survival factors for plasma cells. So particularly the TNF family member APRIL, in conjunction with BAF, IL-6 are known to be soluble mediators of plasma cell survival, and cell contact molecules, including CD28, are expressed on the plasma cells, and CD80 from support cells are also survival signals that feed into the cells. So in this system, one can now imagine that the localization of an antibody secreting cell will be sufficient to ensure its persistence. And in this way, you could equally conjure up these two states of short-lived cells that don't gain access to such a niche, and long-lived those cells that do. And if they're in the niche, then they're going to be there for good. But of course, there are issues surrounding that need to be explained. So one is that, do all of the cells that reach niches actually live at the same amount of time? Is there any death within the cells that are in the niches? And how does one explain new specificities gaining entry into the long-lived compartment if the niches are all filled? So these are sort of foundational questions, if you like, which have underpinned work that's been done by our group and many others in the last couple of years to try and answer those problems. I have a slide here which indicates that eosinophils may or may not be critical in this process. That particular point seem to have taken a severe dislike to eosinophils in this scheme and have decided to remove them. So how does one do that? How does one study long-lived plasma cells? I've shown you the Bloom reporter system is a fantastic means by which to identify these cells, and there are other systems which have developed that have the same outcome. But that doesn't resolve them according to lifespan or longevity. So in order to do that, we took the approach, as did several others, and these groups are listed on the bottom left. There's been a little small tidal wave of this process in the last few years in this particular system. So if one has a collection of plasma cells and one can initiate a change in those plasma cells at a particular time, so introduce a timestamp, then by following the persistence of time-stamped cells from that initial period, one can accurately measure the decay rate of the population, identify those cells which persist, and compare them from those which have a shorter lifespan, or identify their unique attributes, and in that way perhaps get insight into the process and the mechanisms of plasma cell persistence. In our case, we took advantage of the engineering that Steve Nutt had done all those years ago, but rather than putting in just a single color reporter, we put in a tamoxifen-inducible form of Cree, and we crossed it onto a mouse which has a floxed allele of MCL1. And I've already mentioned to you that MCL1 is critical for plasma cell survival, so this is relatively risky, but heterozygosity for MCL1 is perfectly benign in antibody-secreting cells. So deletion of MCL1, which would occur after tamoxifen activation of the Cree, transposes a human CD4 into the MCL1 promoter and puts that on the surface. And one of the advantages of using the MCL1 is not only that it allows this genetic manipulation, but it's also highly expressed in all antibody-secreting cells, allowing us to get, which I think will be in the next slide, relatively good labeling of all the antibody-secreting cells by tamoxifen exposure. So this is the antibody-secreting cells identified by the fluorescent reporter in the targeting construct and CD3138, so shown in each bracket. And below is the degree of timestamp labeling we achieve on those antibody-secreting cells in the bone marrow and the spleen by, in this case, 2 sequential daily injections of tamoxifen. So effectively, we're able to label all of the, well, 95% of the plasma cells in the animal in that 2-day interval, and then follow their decay. So we did that, which is what's shown here, and the blue circles are our data for the representation of label cells at time after labeling, shown for, in this case, the bone marrow. And one can see the decay curves that we were able to model, and the modeling was done by some colleagues of ours, Phil Hodgkin and Mark Dowling, who are expert in this process. We looked at 2 models. One was a dichotomous model, which is shown on the left, and that's in which case there are 2 decay kinetics operating within the population, a rapid and a slow. And in the continuum model, it's a single kinetic that describes the entire population. There's a slight preference in the modeling world for the dichotomous model. But in reality, it's actually extremely difficult for us to distinguish between them, because the lifespan of a mouse is not really long enough for us to resolve cells that effectively have an infinite life, which is the property in the dichotomous model, versus those that have very, very long-lived lifespans in the continuum model. But either way, the end result is that we are able to describe the rate of decay of the homeostatic populations of plasma cells in different tissues and at different ages. And we could measure that and show that it increased with the age of the animal. So it was more rapid in younger mice than it is in older mice, and that it was longer in the bone marrow than in the spleen, more or less as expected. So in our particular case, we didn't find any difference for the decay rates of different isotype antibody-secreting cells in the bone, whereas some of the other groups who have done these experiments in around the same times have found such differences. I can't explain that at the moment, but there are some interesting differences in the different models that may reflect the environment in which the mice live, and that itself is, I think, an interesting point that will need to be resolved. So basically, we have a system by which we can describe plasma cell decay, we can identify long-lived antibody-secreting cells, and we can do that with good efficiency and in multiple tissues at multiple ages. So what can we tell about the cells that are long-lived compared to those that are short-lived, now that we can actually identify them as having that specific attribute? We can actually put a value on that parameter of lifespan. Well, one thing we looked at was the variable gene usage and somatic mutation. So you might have surmised from the scheme that I showed at the beginning that long-lived antibody production was a property of cells that came out of germinal centers. Well, in our particular case, where we partitioned the cells as plasma cells that have been alive for less than 63 days or more than 63 days, so less than 63 days are human CD4 negative and more than 63 days are human CD4 positive. So that's the time between the timestamp and the assay. But there was no difference in the level of mutation in those populations. So persistence wasn't associated with increased degree of mutation at the outset, nor was it associated with a particular VH gene distribution. So we couldn't find what looked like either affinity-based or specificity-based segregation into a long-lived and short-lived compartment. There were differences in cell surface markers depending upon the age of the plasma cells. So shown here is, for example, beta 20, which is already known, but confirmed here to be expressed at very low levels on cells which are long-lived, in this case, at least 10 weeks of age, is shown in the red versus the total population in gray. CD19 is a significant population of downregulated CD19 amongst the cells that are persistent, more so than in the total population. CD93 goes the opposite direction. More of the long-lived cells express CD93 than in the total population. Syndican or CD138 was interesting in that while all antibody-secreting cells, or the vast majority, express it, what we found was that in those that are persistent, they have upregulated and are aggregating at the higher end of the distribution of expression of syndican, of CD138, suggesting that it may provide either some advantage or just be a natural consequence of their persistence. So we can see changes. Clearly, we not only did flow cytometry, but we also did RNA sequencing and found that there were indeed genes associated with persistence. So shown here is the heat map of those genes that differed between the plasma cells that were persistent and those that were not. And we had two different points of persistence. So we analyzed these or isolated these cells for sequencing at either 60 days after the labeling. So we had plasma cells that were a maximum of 60 days old, shown here, or a minimum of 60 days old. And we did it in another group in which we looked at the plasma cells that were a minimum of 270 days old. And what I think you might be able to see is that there are clearly differences in this array of genes in their expression between those plasma cells that are relatively young and those that have much greater age. But once they have that attribute, which is apparent here in cells that are 60 days or more, it was relatively stable out to even those cells that were much, much older than that. There wasn't any greater enrichment for a different set of genes that we found in the cells that were long-lived. So if one was to put it in this kind of trace, it appeared that the program that was associated with persistence was established relatively early and then kept on. Even though, as you'll remember from the decay curves, very few of the cells that were present at 60 would still be persisting at day 270. So it's not as if there's, as I said, unique distribution of genes associated with that very, very long persistence rather than some sort of minimum value. And we don't as yet know exactly what that turning point is when one plasma cells would flip from this gene expression pattern to this one. That's something that's currently under investigation. We did this also by single cell expression, and we could identify clusters, which is an attribute, I think, of all of these studies. But in our particular case, we didn't find that longevity was associated with any particular cluster, although some were excluded, such as these, which were known to be plasmablasts. So in our hands, at least, there clearly is a genetic signature associated with persistence beyond some minimum value, and in our case, that was about 10 weeks in a mouse. Within that, it looks relatively diverse in terms of the array of genes that any individual cell would express, which either means we just haven't done enough cells or the right time points or time differences, or alternatively, that there are potentially multiple ways that is able to be sustained as a long-lived plasma cell that might depend upon the individual circumstances or the precise circumstances at the time at which we take the samples for sequencing. Okay, so a little bit now on the population biology. So two of the markers that we found to be different on the long-lived cells, or the cells as they matured, were MHC class 2 and SLAM F6. And we were able to distinguish these three populations based on that, where there's a group that express only SLAM F6 and express neither. And what we were able to determine was that lifespan increased with the decreased expression of both of these markers. So the cells expressing both contained were predominantly short-lived. Those expressing only SLAM F6 comprised a longer-lived fraction, and those expressing neither were the most enriched for persistent cells. And that's what this data shows as we map the persistence of timestamp cells through those compartments over an extended period. So that allowed us to use this as a way of following plasma cell development and persistence without having to do always timestamping. And we use that to look at the ontogeny of these compartments in mice at different ages, so 3.5, 5, 14, 22, and 34 weeks. And what I think you can see is that it takes to about 14 weeks to establish what will be like close to the homeostatic distribution of these markers and compartments in bone marrow plasma cells in mice. So quite some considerable period is required for that to develop. Using the fact that we could induce the deletion of MCL1, we made these mice homozygous for the flox allele of MCL1, did the tamoxifen induction in our timestamping system, but now, of course, we're going to kill all the antibody-secreting cells because we're taking away both alleles of MCL1. But then we examined the repopulation of the plasma cell compartment at intervals after that deletion to see how long it took for those cells to reconstitute the three compartments that were present prior to depletion, shown by, again, MHC class 1 and SLAM F6. So the double positive, single positive, and double negative before deletion. So 1 week after deletion, so as little as 7 days, all of the antibody-secreting cells that recovered expressed both markers. So there were these relatively, what one would consider immature compartments. And as time progressed to about 8 weeks, we had, again, the restitution of the initial or homeostatic compartments. So despite these being in adult mice, they still took about 8 to 10 weeks to restore the homeostatic age-related compartments in these mice. So it was a property of the age of the plasma cells rather than a property of the age of the mouse. It wasn't that old mice generated immediately this compartment of enriched for long-lived cells. They have to go through the same process as we saw initially. Okay, so plasma cells survive by sitting in niches and by receiving signals from outside them. And that niche sustains the plasma cells by sustaining expression of MCL1. Despite this, and despite in order to survive, they have to sit in niches, we see that there's differences in lifespans. And despite the fact that all the niches are filled, we're able to get new specificities into the population. So how does that occur? A mechanism for turnover, or a model of turnover, was provided by Andreas Radborg, who, as I said, developed very much the ideas around this niche and its role in plasma cell persistence. And in his proposal, in taking an interpretation of it, and I don't mean a verbal Andreas on this, but it's really to set up the experiments we're going to look at in a minute. New antibody-secreting cells have a capacity to displace or dislodge existing ones and take their place in the niche. And that displacement separates the older plasma cell from the survival signals which result in its death. So it has turnover, but while retaining homeostasis, which is determined by the frequency of the niches. So if that's true, then if that's the basic format by which this works, then one should be able to alter this by increasing or decreasing the production of new plasma cells. That should change the rate of turnover of the established population. So we have a system by which we can look to measure this. If one was to block the production of new plasma cells, one should stabilize the existing population. So we did this by depleting all of the B cells in our mice after time stamping by treating them with rituximab. So we hope to diminish plasma cell production and then measure what happened to the plasma cells that were present prior to the depletion of all of the B cells. So this is just data to show that we were able to deplete the B cells. We reduced them by 1,800-fold in the bone marrow, 100-fold in the spleen, and didn't affect any of the other cell types that we counted. And then we looked at the antibody-secreting cells. And I apologize for the format of this data, but the red triangles are the important one. And this is bone marrow on here, and it shows the frequency of plasma cells persisting over this 43-day interval after time stamping. And in this case, it's the same initial state of time stamping, but then the B cells have been depleted. So despite the loss of all of the B cells in these mice, it's made virtually no difference to the persistence of the plasma cells that existed prior to the B cell depletion. And that's also true in the spleen. There's a twofold reduction, if anything, in the persistence of cells in the absence of B cells. So we would have expected that these would be more persistent and probably sit closer here to this line, which was their frequency at the time we did the B cell depletion. So in this case, where we reduced plasma cell production, it made no difference to turnover. And I'll just skip through that. But what about the opposite if plasma cell production is increased? And this we achieved, that should be, if you like, in this same scheme, drive greater turnover of established plasma cells. So in this particular case, we achieved this by doing time stamping on a cohort of mice, irradiating them, and then reconstituting them either with wild-type bone marrow, so they'll just have normal plasma cells replacing a normal frequency, or with bone marrow from BCL2 transgenic mice, where the BCL2 is expressed within the B cell lineages, and that is known to result in an excess, a vast excess of antibody-secreting cells. So this would be a circumstance of increased competition, and maybe that would influence the rate of turnover. So this is the frequency of antibody-secreting cell arising from the donor bone marrow, either wild type here or the BCL2 transgenic in the bone marrow, wild type or BCL2 transgenic in the spleen. So a vast excess of plasma cells in the spleens of these transgenic mice, twofold excess in the bone marrow. So did that change the representation of the pre-existing plasma cells with this increased competition? And the answer was it didn't. So this is the persisting plasma cells unchanged, irrespective of the increased competition in the bone marrow, and unchanged here in the spleen, despite this vast increase in competition in that location. So again, despite hugely increasing the frequency of plasma cells that are potentially looking for niches, we weren't able to measure a difference in the persistence of the cells that existed there. So just to get towards, to race through the last little bit, this previous experiment suggested to us that actually what drive turnover was not pushing out by or displacement, but rather intrinsic death of the plasma cells themselves, and that created a vacancy. And we thought if that was the case, then we would probably see something like that if we were to look at immunization. So in this case, and I'll just go quickly through this model, what we did was to immunize mice with vaccine type antigen, which is our favorite antigen, nitrophenol coupled to KLH. And then with different groups of mice over a period of 56 days, treat them with tamoxifen to label all of the antibody secreting cells existing at the time of the treatment, and then let the mice sit out to a common endpoint, which was 70 days. So in this first group, after immunization, we did the gavage 3 days later, and when we killed these mice, or sacrificed them, I should say, at 70 days, the plasma cells in there would be that are human CD4 positive would have to be at least 67 days old. In the next group, they'd have to be at least 63 and 56 and so on and so on and so on. So we should actually see this graded increase in representation of plasma cells as the life goes on, if there's going to be continuous recruitment. So to put that into a numeric format, if there's continuous recruitment, we should see a accumulation of human CD4 positive plasma cells persisting as we get closer and closer to the endpoint of the experiment and further away from the immunization. If, on the other hand, there's a wave of plasma cells that comes out and engages in displacement of existing ones, we'll probably have nothing for a while, and then we'll see an influx of labeled plasma cells that persist through to the end time point. So in doing this, the result we have is, again, quite clear that if we look at our antigen-specific plasma cells, which are shown on this right-hand side of the dot plots, and whether they're human CD4 positive or not, which is shown in the upper panels, I think it's reasonably apparent that there is a continuous accumulation of these persistent antibody secreting cells after immunization in these mice. And indeed, if we plot it out, we actually find it's really linear and suggesting there's a constant rate of accumulation throughout the immune response for plasma cells to persist in both the spleen and the bone marrow. Again, in our opinion, consistent with the idea of those cells taking up occupancy of niches that are vacated at that same constant rate, which is reflected by the gradients of these lines. So this just talks about the affinity of these cells, that it continues to increase, and I apologize for racing through this. But there's an important implication for this, and I think one is that if there is this continuous output and recruitment, then a germinal center of a certain duration will produce antibody secreting cells for the duration of that property, and they're able to be recruited, but they'll be reflective of whatever the degree of mutation is and affinity maturation during that interval. If one could extend these germinal centers over a much greater interval and recruiting more precursors and having greater degrees of mutation and distribution of specificities, of affinities, then it's easy to conceive from that how one could accumulate in the bone marrow compartment plasma cells of considerable, a greater number, and potentially much greater affinity. So in terms of vaccine strategies, I think our suggestion would be that a longer germinal center response is going to be much more beneficial for recruiting a larger number of long-lived antibody secreting cells than a scheme which might have maybe even a higher magnitude, but a much shorter duration, which may be achieved by repetitive boosting at very short intervals. But that's, again, to be tested. So there are some real world implications, if you like, from this. So our data are consistent with a system in which existing plasma cells die spontaneously, and then their vacancies are taken over by newly formed plasma cells that happen to be in that vicinity at the time or during the period at which that niche is vacated. But there are some things which it doesn't explain at all. And amongst these is why different specificities, chief amongst these are why different specificities have different lifespans. And this is some beautiful data from Mark Slifka, published many years ago now, but it shows that in humans immunized, vaccinated with measles, the decay of that antibody, which is reflective of the decay of the antibody secreting cells, has a half-life of 3,000 years, I mean, impossibly long. Whereas tetanus, on the other hand, which we're all familiar with because we get boosted regularly, has a lifespan, a half-life of 11 years. So since that reflects plasma cell production, it clearly suggests that there's different lifespans with different specificities. And that doesn't fit with anything that I've been describing today, which is all about a continuum and a population showing a relatively uniform, or sorry, falling on a single distribution of decay. But and how would one go about achieving this, like selecting cells, plasma cells into this compartment, for example, to have this attribute rather than this attribute? This is taken from one of Mark Slifka's reviews, and in his opinion, it's all about the antigen. So antigens of a particular format, or characteristic, or environment in which they're delivered are able to induce in the antibody secreting cells during their formation the capacity for them to be ultra long-lived. And so one would have these different lifespan characteristics based on specificity. And in his case, I think he would say that the continuum that we see is reflective of just simply the range of specificities that has been used to generate the plasma cells that we're measuring. So that would be entirely within the antibody secreting cells. But there are options to that, alternatives. And I'll just show you some data that we have from immunizing mice with MPKH again. And we follow them here, identify them, and then we can measure their characteristics of MHC class 2 and SIMF6, those components that determine plasma cell lifespan. And the NP binding cells are shown at the bottom. And at the beginning, they all have an immature phenotype. And then that matures out to day 35. And then by day 100, all of them sit within, those that persist, sit within this long-lived compartment. So it's not 100% ironclad, but it's suggestive to us that actually the cells acquire the attributes of longevity with that persistence. It's not that they're born long-lived, they're born with all those features. It doesn't say that they're not born without the potential, but they certainly don't come out ready-made as long-lived cells, suggesting that it may be more of a developmental program and process rather than a strict formatting at the time at which their cells are made. So in what's my last slide, I was going to say, well, that makes us think, well, how is it then that plasma cell lifespan might be determined? And our suggestion would be it's going to be a combination of factors. It's going to be things which are properties of the plasma cells and properties of the niche. So the niches don't have to be uniform. You saw it's relatively amorphous at the moment, very poorly defined. Cells relying on the frequency of hematopoietic cells that are present in the bone marrow. They could be very different in their frequency at any particular point or any particular location. So that could be differences in the cell types of the niche. The factors that they secrete, the concentrations and the availability of those factors will differ. The availability of metabolites and hypoxia is also thought to be one of the key determinants of plasma cell persistence. So all of those things could vary across an organ and between niches, which might make some of them good and some of them bad. The plasma cell itself could vary based on its antigen, its isotype. We didn't see that, but others have. Its origin, so whether it's germinal center, extrafollicular, T independent, B1, and so on. Its adhesion capacity, so its ability to stay stuck to the cells of the niche. How well it migrates to that location. Its metabolism, its signaling, how well it processes the signals that mediate survival. And of course, gene expression and epigenetics, which could be part of that antigen mandated program that they achieve. And I would say to you that our thought would be, our theory would be at the moment, there's a combination of both of these things, and they could be which will determine lifespan. And so those super long-lived cells that Mark Slifka identified in the case of measles could be the optimum of this program meeting with the optimum of this program. And so the combination of those two things create this unique set of factors that allow for those cells to persist for that extraordinary time that pushes them way out on that distribution. But hopefully with the tools that I've shown you and the approaches that we and many others are taking, we're now in a position where we may be able to answer these questions. So I'll finish there, and I'll just close by thanking and acknowledging the fantastic people, whoops, sorry, too far, who've done this work. All of the plasma cell work was done by Zoe Ding and Marcus Robinson, who are sitting furthest away at this table from me. I think that must have been during the time at which we were writing the paper. We're back on speaking terms now. It's all good. And others have helped. Graduate student Jesse Mulder is now working on this, and great technical assistance from Christy O'Donnell. And our modelers were Mark Dowling and Phil Hodgkin, and Rosie Webster was another key contributor. So I'll stop there, and thank you very much for your attention.
Well, thank you very much, David, and thank you in particular for delivering this great talk in the middle of the night, which is quite a feat. So it just remains for me to thank you and all of the attendees. Remind all the attendees that you can ask questions following the instructions that are now depicted on your screens. And thank you all for your support of Global Immunotalks, and we hope to see you at the next one of this series. Thanks again, David.
No, thanks very much for coming. Bye, everyone.